All right. TGx-DDI biomarker. Who doesn't want to hear about that? I'm really excited to tell you about the progress that we've been making over this past year with the TGx-DDI transcriptomic biomarker. It's been a really good year for us. Can we go to the next slide, please? And I'll talk about the team first of all. So I have the honor of presenting on behalf of the team, and Dr. Heng-Hong Lee is one of the chairs of this team. So I hope she's on, and maybe she can answer some questions as well. We have a fantastic team. I would, argue that our team is like the Energizer Bunny. We just keep going and going and going. This project has been going on for over a decade now, and we are excited because we continue to add new partners every single year. So next slide, please. You can see the partner list there. The team is focused on the problem that's summarized in this slide. So I have a summary here of the standard tests that we use for chromosome damage and mutation analyses, and at the top you'll see the in vitro tests. These in vitro tests have served us very well. They're very sensitive, but they do lack specificity, and this leads to a high rate of positive results. People call these false positives, misleading positives, irrelevant positives, but these in vitro assays lack specificity. They're also generally not that efficient, and they don't provide information on the mechanisms causing genotoxicity. And so there's a need for high throughput genotoxicity tests that provide mechanistic information in human cells. Next slide then is what the group came up with over a decade ago as the solution to this challenge. And that's the development of a transcriptomic biomarker that can provide mechanistic context to responses to genotoxic agents. So the TGx-DDI biomarker was developed using human TK6 cells, and these cells were exposed to 28 different prototype DNA damage inducing DDI or non-DDI agents. And then machine learning approaches were applied to extract 64 genes. And you can see how those look on the left-hand side of the screen. 64 genes that allowed us to bin chemicals into the DDI profile or the non-DDI profile. And some of those critical early publications are shown here, so you can see when the project started and its evolution since then. So next slide, please. We are excited about transcriptomic biomarkers, and we have a long-term vision for how they can be used. As you are very well aware, transcriptomic biomarkers, you can click through, Connie or Chrissy, whoever's driving here. These transcriptomic data sets are really complex and difficult to interpret. And so what we think the value of transcriptomic biomarkers are that you can sort of efficiently and really non-subjectively extract information from a complex transcriptomic data set. And these transcriptomic biomarkers can be aligned against adverse outcome pathways to enable pretty quick and accurate identification of a hazard or a mode of action. The group has also been taking it to the next step. It's not just about yes or no binning into hazard or mode of action. It's also about the dose response. So understanding how the genes in our transcriptomic biomarkers respond to dose to define thresholds of toxicity, and even maybe develop ideas for using these for transcriptomic points of departure. And so in the end, after we've done, the in vitro to in vivo extrapolation, we understand the human exposures, there's a variety of different contexts of use that we can apply these in for different decision-making contexts. So the next slide is what we decided should be our applications in our 2017 paper in PNAS led by Heng-Hong. On the left-hand side is addressing that problem of the low specificity of the in vitro assay. So when you have a positive chromosome damage response, but you have a negative Ames assay and a negative in vivo micronucleus test, we suggest that you use the transcriptomic, the TGx-DDI biomarker to help you determine if these are relevant or irrelevant positive results in that in vitro chromosome damage assay. So here we're using the term relevant and irrelevant. And we have this fancy three-pronged statistical analysis that we applied that is built into a standard operating procedure that allows you to make these very concrete decisions, non-subjective decisions about whether it's a DDI call or a non-DDI call. And over on the right is more my area of expertise, which is environmental chemicals. And there we can use the TGx-DDI biomarker in a variety of different contexts of use. So hazard ID, prioritization, mode of action analysis, weight of evidence, and potency assessment. So these are summarized in the PNAS paper in 2017, and the context of use on the left is the one that we have brought forward to the FDA's biomarker qualification plan. And I'm going to tell you all about our progress with that towards the end of the presentation. Next slide, please. We've undertaken all kinds of validation work, and those of you who've been on the eSTAR committee for years have heard about this. But now we're up to well over 100 chemicals that we've tested across 5 different transcriptomic platforms. We've done cross-lab validation. We've got 2 different cell types that we know the biomarker works in, TK6 cells and HEPA-RG. And here I'm just showing you the highlights of that 2017 paper. That was 42 agents that were part of a test set that we tested the TGx-DDI biomarker in. So in this study, the agents were characterized as either belonging to class 1, which are direct acting DNA damage inducing agents, class 4, which are negatives, and class 5, these misleading or irrelevant in vitro positives. And at the top, on the top right, you can see how the agents that we picked performed in the Ames assay and the chromosome damage assay, with yellow boxes being positive and blue being negative. And you can see that TGx-DDI did a great job in terms of high accuracy in identifying class 1 and pretty good specificity as well. In fact, it effectively classified almost all of the class 5 agents as negatives. Next slide, please. And then we also need to understand then, now we've got a lot of validation studies, we went on to ask, how would we use this in different decision-making contexts? And so we've been undertaking different case studies, some in partnership with regulators. Here's an idea of a few of them. There's more than are listed here. In the first one, we looked at caffeine, which gives positive results in the chromosome damage assays, and we showed that caffeine is negative by TGx-DDI. In the second, we worked with the Health Canada's New Substance Assessment and Control Bureau, and we looked at the concordance of TGx-DDI with the micronucleus test, and provided new information on 2 data poor chemicals, benzene triol and disperse orange. And we saw high concordance in terms of hazard calls as well as potency using these assays. And then at the bottom, we have a more recent one where we looked at 3 drugs with questionable genotoxicity finding. And here it's an integrated testing strategy with the high throughput COMET assay, demonstrating how these platforms can be used together. And this also had a bit of cross-platform, a bit of cross-lab analysis going on in that case study. And then the next one is a newer one. Next slide, please. This is out this year, led by Anne-Marie Fortin from Health Canada. And this is how we've integrated TGx-DDI now into Health Canada's GeneTox 21 program, led by Dr. Paul White. So in this paper, TGx-DDI was used in an integrated testing strategy with the multi-flow and the microflow test to make decisions about the genotoxicity of 10 data poor compounds. You can see them over there on the right. And it's a really cool paper. I hope you'll take some time to take a look at it, with a neat data interpretation workflow that shows really cool integration of the transcriptomic analysis by TempoSeq of TGx-DDI with the multi-flow assay available from Litron Laboratories, which detects gamma H2AX and p53 phosphorylation and other endpoints. Really, really nice integration with that. Next slide, please. So one of the things that's been extremely rewarding to be part of a committee, a working group that goes on so long, is to see the impact of your work and its adoption around the world. And I would argue that I haven't been to a GeneTox meeting in a very long time, in a few years now, a couple of years, where I haven't seen at least one presentation about TGx-DDI by somebody who's not in our working group. And so that's been extremely rewarding. And one of the really exciting things that happened was that the international workshop on genotoxicity testing, the international workshops on genotoxicity testing that were held in 2022 in Ottawa, decided to have a whole working group focused specifically on transcriptomic biomarkers. And of course, TGx-DDI featured very heavily here, and was really used as case study materials for this group. So watch for this paper coming out this year, and I want to read the consensus statements, because the consensus statements are based a lot on the work that was done within our working group in HESI. So TGx biomarkers for genotoxicity can be used. This is the consensus opinion of people from across the world, can be used in weight of evidence evaluation to identify potential genotoxic mechanisms and hazards, to identify misleading positives in vitro genotoxicity assays, and as part of integrated testing with standard batteries of genotoxicity tests or other NAMs. And the second bullet is that several biomarkers, that includes us, have been developed with sufficiently robust training data sets, validated with external test sets, and have demonstrated performance in multiple laboratories. These biomarkers can be used following prescribed study designs and models designated through existing validation exercises and weight of evidence evaluations. So fantastic that the world agrees on this now. So I want to give a shout out to Roland, who was the working group leader here, and to Chris, who was the co-chair, and Hang Hong, and Andrew Williams and others. The group was full of TGx-DDI working group members, and we've had quite a nice impact there. Next slide, please. And the other thing that I just want to highlight to those who are new to the program is that there has been a little bit of trepidation about the use of transcriptomic biomarkers because of the complexity of transcriptomic data analysis. And in collaboration with Jennifer Fostel at the NIHS, there's a tool that makes this analysis amenable to anybody. You don't need to be a bioinformatician to use it. And we're using this as part of the FDA validation exercise now. And in fact, it's straightforward enough that I'm now teaching this to students in my fourth year class, and they can do an analysis in just a few minutes of TGx-DDI responses. Next slide, please. So it's been a long ride. One of the things that I wanted to shout out about this year was over on the right, you'll see there's a CRO who now offers this as a service. So Cytovation, led by Les Recio, now offers analysis of HEPA-RG cells with TGx-DDI and micronucleus assay integrated as an integrated test strategy. So it's very great advancement here. So labs around the world can contract out this service to others. So you can see all the progress that we've made. And now I know, and it has been a roller coaster ride. I say this pretty much every year. But now I know you want to hear about the FDA biomarker qualification process. So the next few slides will tell you about how we're doing here. So we were restarted when the program restarted with the FDA, and these are all the steps that we've already accomplished. We've done the letter of intent, we've done the status report, and we have developed our qualification plan, and it's approved and it's underway. We are grateful to the FDA for all of the feedback that we've received. And one of the things that they felt missing in all of this data that I described to you was actually a ring trial. And so that's what the biomarker qualification program is about now. And we're conducting those final validation studies, and we're very close to being able to submit our full package now. So next slide shows you, we needed to get find money in order to do this ring trial. And a big thanks, a huge thanks to the HESI staff who were really critical in putting together an FDA U01 grant with the team that was awarded $250,000 to do this ring trial. And so you can see what that funding is supporting within the ring trial. So the next slide is the actual validation study that we're undertaking. On the left, you can see our qualification plan that reflects years of conversation and input, years from the FDA biomarker qualification reviewers. And what we wound up with was this ring trial, the objectives of which are to assess the cross-laboratory reproducibility of TGx-DDI on one platform. NanoString was the selected platform at 4 sites. and for 14 chemicals, plus I think there's 3 or 4 controls in there as well. So you can see a little bit of the experimental plan here. And the big thing, the FDA didn't want the same old cast of characters running the assay. So this is, if you go to the next slide, a whole new injection of team members into our project. You can see them listed here. So a big thanks to these people who've come in now. You can see their roles from Procter & Gamble, Sanofi, Burleson Research Technologies, Children's National Genomics Corps, and the Wistar Institute Genomics Corps, who are undertaking this ring trial. The next slide, please, then, is to show you our progress. We are scheduled to be finished our ring trial by the end of the year. We're making excellent progress. You can see most of the labs are finished, almost everything, and we're just really going to be looking at the concordance between the labs for these labs, probably in the end of November or December. And so we're very much on track to finish when we were striving to. Next slide, please. And in the meantime, we have been out on the stage telling people about TGx-DDI and encouraging adoption. So you can see Connie on the left presenting at tomorrow's data today, and then there's me over there on the right speaking about the TGx-DDI at the World Congress of Animal Alternatives. The next slide, please, then, is our timeline to bring this project to a close for the trial. So as I said, we're hoping to have everything finished up by the end of December, at which point we'll submit a preliminary report to the FDA, and we'll begin writing our full qualification package, and we'll be writing our manuscript then. And so sometime in 2024, we expect to have all of that submitted and be waiting for approval. And in the meantime, at that point, we will be thinking about the future, because like I said, we're the Energizer bunnies. We just keep going and going. But some of the things we're talking about, and we hope that others will be interested in, include things like actually developing an OECD guidance. There's already the GenoMark biomarker. So is it time for us to have a TGx-DDI guidance document? Or going into the OECD through the use of an integrated approach to testing and assessment, or developing a defined approach. These are things to be determined. We're thinking about hosting a workshop to discuss our qualification path so that others can learn from our experience. And we want to start pushing. Of course, the FDA is pretty focused on a specific context of use. We'd like to start pushing the utility of our biomarker a little bit further beyond just inclusion in weight of evidence evaluation. So I think that brings me to the end of my presentation. Is there one more slide? Oh, the learnings, what have we learned? I forgot. Okay, so an important learning was I have personally had never run the NanoString platform. Heng Hong and her crew has had a ton of experience, and Al Fornes and others at Georgetown University have had lots of experience with that. It works very well in their hands. So the labs who were doing this came in new. They hadn't done NanoString analysis before. And it turned out that some of the cores that they were using were also somewhat new to using NanoString. And so there's still some questions that are coming up from time to time about, bumps in the road associated with this being relatively new to some of the people participating in the study. We learned a lot, when you're close to a protocol and you've been doing it yourselves for many years, and then others try and read your SOP and apply it. It's not always as straightforward and easy to interpret as you envisioned it to be. So we're learning a lot, especially about edge cases. We're very glad that we included multiple concentrations in this experiment. And we also realized that when we decided what was success, what we would call success for the outcome of the TGx-DDI biomarker qualification, we might have been a little overly optimistic on our expectation of concordance. We basically said, it's going to be 100% concordant, no worries. So we may want to be having a discussion with the FDA about maybe we were a little overly optimistic there. One of the things that we also learned was about the importance of being extremely clear in our quality control and filtering protocols, and the new team members have been extremely helpful in helping us to figure out how our SOPs lacked a bit of clarity in those areas. And then finally, I just wanted to say, and I do think this is my final slide, that we wouldn't be able to move this project forward without huge encouragement and help and management by the HESI team. So Connie, Chrissy, Cyril, huge thanks to you. We meet twice a month, and we've been doing this for a long time, and we love it, and we are just so lucky to have you. All right, thank you. I guess the group, if I missed anything, please jump in and let us know if you have comments or questions.
Great, fantastic presentation, Carol. Question from Priyanka in the chat. So they asked, Takeda would also like to participate in the ring trial. It's probably a little too late for that, but how else could folks engage?
I think what we need to do is finish this ring trial. And then I really do think that we need to think about the next steps. And I think that's the perfect opportunity for new people to come in. I mean, do we want to do another case study on other contexts of use as a HESI team? Do we want to maybe develop an integrated approach to testing and assessment case study for the OECD? And we could think about, whether that could be used as a mechanism to develop a defined approach or a mechanism for a guidance document. So I think that, once we've finished this ring trial, it's the perfect time for new members to come in.
Great, thanks for that. A few other questions. One from Caleb. He asked, can TGx biomarkers be useful to move away from qualitative, positive, negative hazard classification for genotoxicity and identify quantitative thresholds of genotoxicity for risk assessment?
I love that question. I love it. And we have been doing work on that. So this begins to overlap again with the transcriptomic point of departure working group. And what we have been doing is looking at how we can derive a point of departure from these gene sets. And when we use a very simple approach, and I think this can be refined to identify a TGx-DDI point of departure, we see a very good concordance with the point of departure from other genotoxicity endpoints, like the micronucleus test. We have a really nice study showing that it's pretty much a one-to-one relationship. Same with the COMET assay, we get similar points of departure. We're shifted a little bit there, but similar points of departure. So I think we do need to push this forward, and that quantitatively we can use this to identify thresholds of genotoxicity. And so I think that's another space we should be working in.
Great, thanks for that answer. Question from Patricia, are we able to share the compounds that were tested? I think she means in the ring trial, but maybe the full list. And what about the diversity of the compounds?
So we can't share the compounds in the ring trial right now, but we can certainly share the full list of compounds. We have a table in our FDA biomarker qualification package that summarizes every chemical that's been tested with TGx-DDI from all the labs, from that time point. There's more since then, but Patricia, I can send that to you, and this includes people.
Just to clarify that question, I just want to clarify that question. It was more because in the initial trial, there was a limited set that was used, and I assume they were, especially the type of chemicals that were tested. I assume in the ring trial that new set of compounds were different mechanisms. Meaning, are you amplifying the type of mechanism testing?
They're not part. So the compounds in the ring trial are not part of the training set. Those original training set compounds, they're outside of that. There may be some overlap with the 42 chemicals that were tested in the PNAS paper, the test set chemicals. And they do span a variety of different mechanisms of action.
Okay, thank you. That's what I want to know. Thanks.
Thanks, Patricia. Cyril had a comment back to the comment from Takeda that, what we've heard from the FDA is that they would like to see sponsors use these biomarkers as part of their submission and part of the weight of evidence. So there's opportunities, Carol said the tool's online, there's SOPs in place. So folks being able to use the tool would really support, the uptake and usage of it. Would you agree with that, Carol?
Absolutely. Yeah. I mean, it was a bit of the chicken and the egg problem when we went to the FDA. They didn't want us to begin the qualification process until we could demonstrate that labs were using the TGx-DDI biomarker. And we felt we needed the qualification process to be completed before labs would use it. So, and we are very collaborative. So anybody who wants to use it and needs advice or help, you're welcome to our SOPs. You're welcome to our expertise. We'll happy to provide some coaching.
Great. We have a comment from an unknown user, which is always a bit ominous. So if you would like to elaborate on it, go for it, but I'll read what's there. At least for DDR assays, the concentration difference between carcinogenic responses based on specific markers and frank cytotoxicity can be very tight and close. You label those as edge cases, but this is at least in-house, seems to be more the rule than the exception, excuse me. Any thoughts on proper dose concentration spacing and teasing apart genotoxicity and cytotoxicity?
Yeah, really, really good question. And yes, this is what we're seeing with the edge cases. So with TGx-DDI, we've basically been following the in vitro micronucleus guideline, which sets the cytotoxicity limit at 45% plus or minus 5% viability. Sorry, so yeah. So 45% plus or minus 5% viability at the 24-hour time point. So bear in mind that we sample early for the transcriptomic response. So we have a separate set of cells that we use to measure viability at 24 hours. And that threshold is important. When you exceed it, it does look like we get false positive results. And so, being close to that threshold is really important. And we have moved more towards the TempoSeq platform, which is cheaper and higher throughput than RNA-seq and then microarrays, and Heng Hong is using NanoString. And that allows us to have a nice concentration response. So I fully agree. We need to have, a concentration up that's close to but below that cytotoxicity threshold, and as many doses as you can scale down from that is important.
Great, thanks, Carol.
And I would say that for some of the mechanisms of genotoxicity, it's actually really sensitive, the TGx-DDI biomarkers. So places where, really low concentrations where we weren't expecting to see a response, we're seeing it even down at those low concentrations. But sometimes you have to push it.
Great, thanks, Carol. And the last comment I see is support from Peggy about agreeing we should start on an OECD protocol for DDI in order to have in place once we have regulatory approval.
Great, thanks, Peggy. I think that's important too. It's such a daunting journey, but I think that we have a lot of the legwork done in this FDA biomarker qualification document. So I'm hoping that we can bring this to the OECD and we will be well ahead of the game.
Yeah, and knowing how long it can take for OECD, you don't want to wait too long. But hopefully we've paved enough roads that we'll be able to slide it through quick. I really applaud you for all your resilience on this project, I gotta say.
The whole team stuck it out.
Amazing.
Great, thanks, Peggy. Thanks, Carol. Carol, anything to conclude from you?
From me, no, this is fantastic. Just a really, really, really big thanks to the team. And I don't know if Heng Hong's on, but she's been a champion through all of this too. And I don't know if she wants to say anything.
Trying to see. I know she had one meeting she had to go out for. I don't know if I see the name. Heng Hong, are you there? Oh, you
are. Well, Carol, it's a nice presentation, and thanks for all the audience have all these wonderful questions. And I don't have anything to add on at this point, but I'm here if you have anything about the TGx-DDI or the NanoString pipeline that you want to know more, you can feel free to contact me.
Thanks, Heng Hong. Okay, it's great.
All right. Thanks, Carol.